Dear ladies and gentlemen, my name is Johannes Holz from Hamburg. It's my great pleasure to discuss the role of Thrombinator in my practice. The Thrombinator device is designed to create an autologous activation serum at the point of care. This serum is injectable through a small cannula, is easy to use, is a fully autologous approach, and provides additional growth factors in a matrix to the orthopedic site. In roughly 20 minutes, the Thrombinator device creates enough thrombin serum to activate PRP, platelet poor plasma, or bone marrow aspirate. This is approximately the same amount of time as the ACP Max or Angel system requires, creating little disruption in the OR workflow. Activated autologous fluid with thrombin serum is easy to work with. Keep in mind, this is not a fibrin glue. The clot takes about 15 to 30 seconds to form after mixing with autologous fluid. This works in our benefit as the clot percolates into the surrounding tissue, delivering growth factors over an extended period of time. The Thrombinator system uses a clotting cascade to create a thrombin-rich serum. We can break down the process into 3 steps. The first step is being the activation. The second step is the production step. And finally, the third step is clot formation, as you can see here. In the first step, the Thrombinator device initiates the clotting cascade using beads and agitation to the concentrated factor 10A or prothrombinase. In the second step, we are producing thrombin. We accomplish this by adding more autologous fluid to the system. The prothrombinase is the device produced in step 1, mixes with the added autologous fluid. The prothrombinase cleaves the added fluid and produces thrombin. In the last step, we remove the solution from the Thrombinator device and mix it with PRP, PPP, bone marrow aspirate, or whole blood. This allows our created thrombin serum to interact with the unprocessed blood, which remains rich in fibrinogen. The interaction of fibrinogen and thrombin creates a fibrin-rich solution that stabilizes as a clot. The clot created with the Thrombinator device does not require centrifugation, heating, or additional reagents. It is entirely autologous and stable, as you can see in the bottom right image. Here you can see the Thrombinator serum being applied to a tissue defect arthroscopically. That serum percolates into the defect and mixes with the applied graft while it begins to form a stable clot. In comparison to an allogenic fibrin sealant, studies show that autologous fibrin has a lower density as well as less fibrinogen and thrombin. This may allow cells to more easily infiltrate in the matrix, improving cell migration and matrix deposition. In an ex vivo model, comparing allogenic and autologous fibrin clots, both clots perform similar in terms of graft adhesion. Thrombinator thrombin improved mechanism compared to the allogenic thrombin when combined with PPP fibrinogen. PPP fibrinogen was equivalent to allogenic fibrinogen for mechanical measures when combined with the Thrombinator thrombin. Thank you very much.